Good day, everyone. It's way back in 2019 when I first came here in Auckland, and back then I'm just talking about a qPCR that I have optimized for scabies diagnosis. And this is after I met Dr. Simon in Darwin, because we also presented the optimization of scabies PCR. Shortly after, we established collaborative work with the University of Auckland with Dr. Gerard and Dr. Simon. And of course, along with the collaborative work also came COVID. So they came hand in hand. But despite all the challenges, I would think that since then we've gone a long way in improving the diagnosis of scabies supported by DNA evidence, which is PCR. So let me take you to our journey. PCR, as you know, only came about when we had COVID because it's a technology that we all relied on for confirmation of our infection with COVID. But it is really a fast, highly sensitive, and specific method for identifying disease pathogen in a sample. The basic requirements are, of course, you have to know the sequence of your pathogen in question. In our case, it's the DNA of scabies. It's publicly available now. You know, the genetic material of the scabies mite is now published in the literature. So you just need to look at the sequence, and then you can design your primers. Primers are the ones that start the DNA replication. PCR is basically just making several copies of your DNA that you are targeting, and it's for you to have a look. Once it has many copies, then you will know it's a positive test. And these are just the basic requirements. You need the Taq polymerase, which facilitate the DNA synthesis or multiplication, and you need this nucleotides in the reaction, and of course your DNA that will come from your mite that you have extracted from your sample. And you do it in a PCR machine. This is a small machine. It's like thermocycler, that there is a cyclic that provides denaturation. So you denature the DNA, you anneal the primer, and then you extend it, and it goes several cycles until you see a product that is positive or negative of that PCR mix. So there are many variations of this. The first one that came about is the conventional PCR. This one is you prepare all the mixes, you whack it in that machine, but you need to visualize your result in an agarose gel. And this is, you have to put an ethidium bromide dye in the gel, and you look at it under the transilluminator. So what you see here are positive bands, and these are negative. So this is a negative PCR, and that's a positive PCR. Well, recently, this technique can be tedious because you need to prepare gels, especially like the survey they've done about 400 samples. So you won't do this anymore because it's really tedious, and you can also only amplify a single target with this method. But there is also now the availability of master mixes that you don't need to put all those components that I have mentioned in the earlier slide. You only have to add your DNA and your primers. And also, you don't need to pour gels. You know, you can buy now precast gels, and you can have a look. Another improvement of this is when you look at it's called quantitative real-time PCR. So this one is a significant improvement because the first one, you look at the end point of the cycle, like you make 40 cycles, you get the endpoint PCR in an agarose gel. This second improvement of the PCR technology is you view the amplification or multiplication of your DNA fragment in real time. And it gives you a quantitation value which is equivalent to the number of cycles required to generate a fluorescent signal. And this is how the amplification plot looks like. And this is your cycle threshold, which means that these are negative background fluorescence, and this is where your positive amplification starts. So this is like the threshold, this line here, and this threshold will tell you that this is the signal now of a positive reaction above the background. And this cycle quantitation value also is synonymous to the number of copies, or how many copies of your scabies mite molecule is in your sample. So this gives you also the number of copies. Another variation of this is you need to do a standard curve. This is just like your reference that these values are generated. So this one, you have all this, and you can see that like in COVID, the higher CQ value you get, the higher the number of copies there are in your target. So, and that is establishing your cutoff or limit of detection of your assay. There's another modification of this to make it more sensitive is not just using your forward or reverse primer. This is also adding a probe. This probe identifies your target sequence, and it's usually a long sequence put here in the middle. And again, this is detected by fluorescence detection, and you also get those amplification curve. So, but even high technology stuffs can have some limitations. The real-time probe-based PCR can also be problematic because sometimes you have very low abundance of your targets, and this happens in the real scenario that you don't really get a lot of mite material from your population. And there are also some other inhibitory materials in the sample that can inhibit the amplification efficiency. So we need to circumvent all these limitations. The thing that I've spoken here about is our probe-based qPCR test for scabies. Normally, clinicians will do a scalpel blade, scrape off the lesion, and have a look at it under the microscope. And of course, the early infection will only have, in one suspected lesion, they will only have 1 to 5 mites, which is really low. And I'm talking about a scabies mite here, which is like a speck of dust. You don't really see it. In a little needle that you poke it with, it will just be like a speck of dust on the tip, and this can easily be missed. So what I have optimized is instead use a flock swab. The COVID swab is like your cotton swab, but this is bristled. There are bristles on here such that it increases the density or your chances of getting the mite material. So how it's collected is you dip it in saline, and then you swab the lesion, and you put it in a DNA buffer, and this one is already your transport medium, and you just break off the swab in the junction and then keep that in that until ready for analysis. So you can keep it at room temperature while you're in the field, or keep it in the fridge, or freeze it until it's ready for analysis. And our qPCR for scabies also, we have optimized a way of extracting the DNA. This is a vital step because, first of all, these first two are your vital steps in the success of the qPCR. You need to be able to identify the proper place or the lesion that you need to swab. So it's so important that clinicians or survey people really know how to identify what they suspect are scabies lesions. And another part of our optimization is to come up with a really very accurate and sensitive way of, or optimal genomic DNA extraction, because you have the swab there. So after all the different methods that we have tested, we came up with just using a commercially available kit. So this is, you can buy this from the suppliers, and we found out that this is really optimal for our purposes. And we have designed our qPCR based on probe-based qPCR, and these are the targets. We have this is COX-1. One of our targets is a mitochondrial gene. In the whole scabies genome consists of coding and non-coding region. Coding region means the nucleic acid translates to protein or amino acid sequence, and this is our first target, COX-1. and this is published. Our alternative targets is SSR5 and SSR6. You know, in most helminths or parasites, the non-coding region, the one that does not translate to protein, they are more abundant. So if you are targeting your PCR in more abundant region of the genome means the whole genetic makeup of the mite, you will likely increase your chances of getting more positive result because you are targeting a more abundant region. And this is from SSR5 is a microsatellite, it's called, and SSR6 is in the long tandem repeat. So in the scabies mite genome also there are repeat sequences, but these two both came from the non-coding region. So all in all, we have 3 targets for our qPCR that we subject 1 sample for analysis. So when we were ready in the laboratory, we took this qPCR to check its performance. First of all, we went to Royal Darwin Hospital, and these are hospitalized cases. So these patients come there for really clinical diagnosis of scabies. So they had proper microscopy, they checked, they did scrapes, and the Menzies School of Health Research did the microscopic examination for us. The clinicians used the dermatoscope to really look if it's scabies case. So you would need a well-defined sample initially to check if your PCR really works. And then we also collaborated with Darwin Dermatology, and this is to check the specificity of the assay. People come to Darwin Dermatology for non-specific skin diseases. We had cases that had eczema, allergic dermatitis, and we had fungal infection. We have bacterial infection cases there. So all other cases and not scabies. And this is to have a specificity check of our newly optimized assay. So this is the first, the amplification curve of our 2 targets to start with. We have this standard curve or our positive control, and at that time our limit of detection for both assays were 3 copies of the target in every reaction. So we are limited by 3 copies, so the sensitivity was not that high enough. So we also tested the specificity. As you can see here, we tested our assays on house dust mites, other closely related species like the head lice, staph and strep, and we also had these samples collected from patients with other skin conditions, and you can see here that it's 100% specific, so we did not detect any positive result in any of these other skin conditions apart from scabies. So after that, now we are ready to take it to the field. So we went to Kimberley, and then we collected samples from children from scabies endemic communities. So we got luckily 15 samples, but our positivity was only 3 out of the 15 collected. And when we look at the CQ values, actually initially they were all negative, but we saw the amplification curve, the fluorescent curve in the qPCR. So we ended up modifying the curve, our reference curve. Instead of a 10-fold dilution here, from the 6th dilution and onwards, we did 2-fold. And this is now the limit of detection of our assay now is less than 1 copy per microliter. So if you go back there, true enough, the ones we collected from the children were really less than 1 copy. So now we can catch those really lower copy number of targets in our PCR. So we have progressively improving our methodology so that we really catch the lowest of them all of the number of copies that you can get from field samples, because it's totally different when we went to hospitalized cases. But field samples are, you have all the inhibitors, you have everything there, plus the low number of copies that you can get from field samples. Then that's when we have started collaborating with the University of Auckland, and all in all, they have collected 64 that we have tested for our probe-based qPCR, and out of those we get 15, and that's 23%. But of course, it also followed our previous observations that the values were high and the copy numbers of those targets are usually less than 1. So it's 0.9 copy to less than equal to, this is the highest copy numbers we got from the samples from the Auckland children. And that is now published in this paper. The results of that has now been published in this paper. So we've made significant improvement. What else can we do so we can increase the sensitivity? So today I'm going to take you to the latest in PCR diagnosis, and this is the use of the most or the newest modification or upgrade of qPCR, which is called digital PCR. Digital in its sense means there's only positive or negative result. You can only go positive or negative, that's it. And all the you still have to prepare your PCR mixes just how we did it before. So we still implement the protocol, except that we use a different instrument now. It's called Qiaquity 1, and this one is capable of multiple targets in one go. So you can query the 3 in all one PCR mix. And this one, instead of using 96 or 384 well plate from our previous protocol, we can now use nanoplates that can do 8.5 partitions, and that is like synonymous to your number of replicates. We do triplicates in our first assay. Now we can do 8.5 replicates of one single PCR mix. And this one does not need a standard curve or reference. You only had to put your positive and negative control and plus your no template control. This is highly sensitive in detecting very low DNA targets because it reports absolute number of copies per microliter. So it works by your PCR mixture, you prepare it. For example, our previous protocol uses 12 microliters. What the instrument does would partition it into 8.5 partitions in the nanoplate, in the new type of plate. It's called nano. And then it measures the end of the endpoint fluorescence after the cycling has been performed. And this technology does not rely on the kinetics of the PCR, so it doesn't bother with your inhibitors. It's less sensitive to inhibitors, and of course we don't need to use a standard curve. The results are shown because every well is imaged, and the results are shown by a Qiaquity software. So there's a software that helps you in the analysis. So this is one representation of the results, and this is showing our 3 targets. The one on the top is our SSR5, and these are our positive controls. So it only partitions your results to positive partitions on the top and negative partitions at the bottom. So this is SSR6, and these are the positive, and those are field samples, and they're negative, and that's COS. So visually, in a 1D scatter plot, you can see that the positive is well separated from the negative results. And this line here is synonymous to your cycle quantitation or CQ threshold. This divides the partitions between positive and negative. The software also gives us results in Excel format. As you can see, these are our controls, and it gives you the concentration or the absolute number of copies per microliter on this column here, and this one is calculated based on the calculation, based on Poisson distribution. So there is more of a mathematical calculation here based on Poisson distribution. So these are the copies you get for each microliter reaction. And because I've used an 8.5K partition nanoplate here, so it tells you if all the partitions are valid. This is synonymous to saying that yes, your assay worked because you are close to 8,005. And these are your positive partitions, and these are your negative partitions. So these are the per well image that's being put in an Excel format. But what you are really interested here are the copies per microliter. As you can see from here, one partition positive, it can detect one partition positive, and the concentration of that in absolute copies is really less than 1. So this is 0.429 copy of the target, and which is my target here. This is SSR5, and that is my copy for microliter. So it gives me a quantitative number of copies of even less than 1 microliter, and that is only evident in one partition. So visually you can see this in each well. You see? When they're all negative, this is how it looks per well, and the software also shows you a signal map. And can you see the single partition here that's positive? This is the one here, the green fluorescence there. And here on this well, there are 1, 2, 3, 4 partitions that are positive. So as low as 1 or less than 1. And this gives you the number of copies in the previous Excel. And this is when all your partitions are positive, and usually this is your positive control. And this could be a sample that have lots of positive partitions, which equates to the concentration or absolute number of copies in each well that you put in your qPCR or the mix. So as you can see here that we have moved to a really highly sensitive and specific assay. Well, specificity, we are on our early stages. We also did the same thing, and COX is our target here. So we did a run of the house dust mites, the head lice, the sheep mite, the staph aureus, and human skin. And you can see here that we never detected any copy in this column here with all the other skin conditions that is not scabies. So again, this is a highly specific assay and highly sensitive, and we are on our early stages, and this is COX-1. We still have to do the other targets, just like how we optimized our first. But just to have a quick check, so we went back to the qPCR that we earlier performed, that were collected in the surveys here. So from the qPCR, we have 23% positivity rate. When we rerun the same samples, now we have increased the positivity rate to 56% using the newest modification or newest technology, which is the digital PCR. So we are continuously improving our protocols and utilizing the latest technology, so we can really nail out the positivity, or nail out the positive versus the negative scabies cases that you encounter in the surveys that they've been sending us. So it's more of, my role is just to really provide a real definitive diagnosis, and I always run this blind. I don't really know the clinical, how they single out. They just send me samples. So this is blind assay using the latest technology. Well, they went to Tonga way back in June when we had the second symposium there, and this is just when they're laying the groundwork. They sent me about six samples, just a handful, and this even got stuck in Australian customs for a bit. We couldn't clear it, we couldn't get it. But when I finally got it, of course, I immediately did the qPCR and they're all negative. So he was a bit concerned. Why are they negative? Okay. Then when I said, I don't know. And then, because DNA can be pretty stable, but as I said, qPCR can also be subjected to inhibitors. And then now when I'm doing this optimization for the digital, the newest technology, let me have a look again. So I've rerun them. So out of the 6, I got 2 samples that were positive. You see, so it's true enough that the digital PCR is not sensitive to inhibitors. So it could just be some inhibitors in the first run with qPCR. And as you can see, I've shown you the partition positive. There's only one that was positive, and that's less than 1 copy, and it's 0.331 copy of this COX target in that PCR mix. So this is really quite impressive that we can detect as low as 0.331 copy of the target per microliter. This, I'm showing you some preliminary results from the last school survey. As you can see there, I have a few 13 samples that were positive, and I'm only using one target. I'm hoping that if I check with the other targets, I can increase also the positivity. But these are just preliminary, and you look at the copies, they're really variable from the Tongan samples, from very low one to some higher copies. So these are all evident in the digital PCR newest technology. So to summarize, I'm not giving Simon and Gerard a magic bullet, but I'm giving them a useful adjunct to help improve the diagnosis of scabies, which is really objective and definitive. But as I would always say, the tools, no matter how high technology they are, they will still rely on your choosing the right site where you collect the samples and some standardization, and you also have to look at the patient's history and all that is really contributing to positivity or negativity of results. In the future, we want to automate the DNA extraction, and we have a machine that it's like a robot that you can do DNA extraction for you, and we have 2 robots in the institute where I work, so we can automate this. So because the current workflow now is there is manual DNA extraction. And of course, I would like to develop a multiplex digital PCR where I can query the 3 targets all in one reaction mix. And this is only just changing the fluorescent dye that you tag your probe. So if you have green, the current one that we detect it is fluorescent dye color green. I can put the SSR5 red, I can put the other one orange. So it's all colorful out there. But my biggest vision, but I don't know if that will still catch up before I put the curtain down or retire. Can I retire? The best thing that I can give all these surveys is a point of care diagnostics for scabies like our rapid test, because we already have the targets, and I'm an external examiner to a PhD student now doing a point of care but for schistosomiasis, another tropical disease, and we have a Queensland company now that can do the work with us. So that is a vision out there. We can be visionaries, right? We can dream. So ongoing collaborative work, we continue to collaborate with the University of Auckland through their Health Pacific project, and they have these 2 projects with Dr. Gerard and Simon and all the people of Tonga and New Zealand. And of course, we can't do this if we have to thank the funders, HRC, the University of Auckland. But on my end, I'm funded by the U.S. Department of Defense, and I currently work a few days at the Australian Defence Force Malaria Infectious Disease Institute, but I continue to get employed by QIMR because they manage the grant. And I'm also working on malaria and arboviruses, dengue, Japanese encephalitis. So all the lessons learned, I've started multiplex digital PCR with the malaria work. It has crossed over now to Scabies. So I learned from the multiple projects that I do. With that, thank you very much.
Are there any questions?
Yes, one from here and then another from there. Go ahead.
Thank you, that's really interesting and really exciting. So we know that the itch that people experience with scabies, that it's a hypersensitivity to the light, and that after we treat effectively, people will remain itchy typically for a couple of weeks afterwards because light still, body parts still in the skin. Do you have any plans to look at how long the test remains positive after effective treatment?
In our experience in Darwin, because they are hospitalized, they remain positive 7 to 10 days with the PCR. But because they've been treated, and the mice material is still there. But it's not synonymous to active infection. Yeah, it's just, PCR is always positive after a few days after treatment. But that's a good question. We can do that study here.
Yeah, it'd be useful to explore the PCR test just in terms of its features, in terms of reaction to treatment and those sorts of things in more detail, absolutely.
Okay, the lady in black and then the lady in, what color is it? Red.
Thank you. I don't really know anything about the immune response to scabies, but could serology be used to diagnose?
I think it has been tried in the past, but there's often a lot of cross-reactivity with house dust mites and other sort of commensal mites that live in the skin. So it's certainly been tried, but I think been disappointing, unfortunately.
Yeah, there's one group at QIMR who has been focusing on that work for so they tried so hard, and still the cross-reactivity has not been sorted. So they dropped off the idea now. So
Yes.
It's okay. So I think it's a really interesting talk. I'm Rachel Newman, infectious diseases physician, but not a clinical microbiologist. But I am really interested in the tests that get very lower level of detection with a digital PCR technique. How far along thinking about clinical validation of that testing method are you and your team thinking about, I guess, false positive tests and whether that testing technique has been applied in healthy controlled populations? And I guess whereabouts is this research PCR currently sitting, thinking about pathways to becoming clinically validated? Just to be very clear that it's not used in any laboratories in our country, I don't think in Australia.
For the qPCR at the moment, Wellington University, so you can have a working qPCR here. At this stage, they're starting to set it up at Simon's collaborators at Wellington University, Victoria.
So yeah, there's some molecular biologists who are working with us on the qPCR there, just to take the pressure off Cielo a little bit. Cielo's really the leading light with the digital PCR. And she developed the qPCR method as well. So, I mean, the qPCR method has had some work in terms of clinical validation, as Cielo mentioned, with the Darwin samples.
And presumably they were people with Norwegian or crusted scabies.
I think the hospitalised
And
ones
Tuesday.
were, yeah, and then she mentioned the Kimberley, which was a community sample of kids with much lower concentration of scabies in that community, which seems to be what we're picking up in our New Zealand, the childcare centres in Auckland.
But the Darwin dermatology also provided us, we've seen the specificity that we have, testing that we've done, so we never amplified any non-scabies material from different patients that have presented themselves to Darwin dermatology. So that qPCR is already proven to be highly specific, and sensitivity we have tested it amongst, but the sensitivity back then when we published it, it's actually there out in the literature, so you can read about it. That time it's only 3 copies, and then that's when we moved on with the Kimberley. That's why we have 2 standard curves that can detect below 1. Yeah, and that's true enough. That's the one type of samples that come from Auckland, that come from Tonga now. So yeah, but the digital, it will also undergo the same rigid validation. So it won't be available for research purposes. Yes, we can do that, but not to implement it as really as a diagnostic tool at the moment. But for viruses, you see, viruses you need to culture. You need to, how long you need to wait for viral quantitation. That one, you can buy kits for viral, and you can easily, get a viral load just by doing the digital. That's the most immediate application of that.
Can I just follow on? So in the number of your positive cases, it was one well out of 8,000 and something wells that was positive.
No, one well divided into 8,500 partitions. So one partition turns out a fluorescent. That's positive.
So is one partition out of 8,000
Yes.
enough to call it positive?
Yes.
It is?
Yes.
And so my follow-on question is then, about half of those cases had one partition out of 8,000 wells.
Yeah.
Was it the same partition?
No. You can see, because it's the software images it. Did you see that some were in the middle, some are on the side, some on the left side? Because the negative, they will all be totally black. There's no fluorescent signal. The signal is being imaged. And then you can see how the negative control is really black. The positive control, positive is I have cloned the targets in a plasmid, so I put that in as my positive control, and you can see total fluorescence. Actually, in the qPCR, you can serially dilute that, makes dilutions, and then you see the fluorescent signal decreasing. Yeah, and then it translates to, I'm saying the absolute number of copies, so that's how it's presented in the Excel format, and that's mathematically derived and following the Poisson distribution statistics. So there is some mathematical derivation of the software for the number of copies that I'm saying, like 0.33 for one valid partition or one fluorescing partition.
So every run is accompanied by standard positives and negative
yes.
controls, right?
Those are your guard that you have. The assay went well, and also you can see if you use an 8,500 nanoplate nanoplate, you have to check the valid partitions. If it's below 8,005, like significantly below, like maybe 6,000 partitions, and then you don't take that the experiment for that well worked. So you have to be close to the 8.5 target partitioning, because it's like distributing the 12 microliter into nano partitions in the nanoplate. So if you didn't get a result So first thing that we look is how many partitions were valid. And that's our clue that worked very well. And of course, you need to check your negative is really negative and positive is really positive.
Thank you. Anybody else with intolerable itch to ask a question? Can we have that? And then, yes.
I might have a wee itch to ask a question. So, thanks for the presentation. Just, I mean, thinking, that ddPCR might potentially be the thing to, I know it's quite expensive and, probably getting around to having available at the clinic might take some time. But it seems like qPCR at the moment, right, seems to be like the easiest thing. I'm just thinking about the difference in the results that you got there. Do you think the targets that you looked for, SSR5 and SSR6, were good targets? And I know you brought up a gel looking at the amplification, right, that was absent in other similar species, but have you just had a look at the sequence, for example, across these different species? Is there overlap? Is there similarities?
Yes. Before we designed the primers and the probe, we collaborated with a bioinformatician from James Cook University. So he did a lot of blast searching and the literature. So the targets, the targets where I chose, he made sure that it did not overlap with any of those species that we have.
Okay.
So that has been sorted from beginning.
Okay. And just that difference, between the two methods, I suppose, when you're comparing qPCR and digital PCR, like, what's your sort of DNA concentration? Are you working with it? How long would you leave it at room temperature before you can expect it to start degrading?
This one, the mix that we use, have all the thermostable polymerases. That needs to be heated up before it starts any reaction. So you can prepare the mixes actually at room temperature because it doesn't get activated unless it goes to the machine.
I'm talking about the DNA as soon as you get the swab, right? With potential DNA samples.
The DNA. Okay.
But
The
you're keeping that
DNA,
at room temperature.
you go to the field, the tissue lysis buffer is the DNA is stable in the swab. In the DNA, it's by QIAGEN. It's called tissue lysis buffer. That is equivalent to the transport medium for viruses. But because it keeps it stable at room temperature for the whole day that you are in the field, and then you take it to the fridge, and then you freeze it when you need to run it all at the same time. So it's pretty stable. Because like the case that it stayed in Australian Customs, the 6, I was still able to, the first time, of course, maybe it got degraded or it had all the inhibitors overwhelming. But with digital, it's not sensitive to inhibitors. So I still got 2 positive despite how many days they stayed in the customs? 2 weeks?
It was a couple of weeks.
Yeah.
So yeah, it's still got positives despite, hopefully it was stored in a fridge. Just to add to all this, I think Oz Diagnostics, we've had some discussion with Oz Diagnostics, who supply lab equipment to Middlemore Lab about a Scabies assay. So they're in the testing phase. And I think the more sort of interest they have, the more likely they're going to have that available sooner rather than later.
Yes. Oz Diagnostics has a multiplex panel of dermatophytes, fungal infections that can coexist with scabies. They want to put the scabies on it as well. So if they can successfully incorporate that, because they're a commercial company and it's published there, I didn't apply for patent. So I'd rather...
Okay, you just need to come back again because there's a lot of interest here. I know you are itching to ask another follow-up.
I was just with Simon, really interested, given that the IX criteria were designed for clinical diagnosis, what you really think about the utility for using them in population-based prevalence studies and
Yeah, well, I think they're
because that's acknowledged in the discussion of the paper as a
right.
limitation that they need to be.
Yeah, well, I think it's like a lot of things. I think it's, what are the alternatives? And I think the IX criteria are actually are probably the best. They're a collective. It's a Delphi process, so there's this kind of idea that many experts had to agree. In terms of this debate, I think one of the things, in terms of, should it be sensitive, should it be specific, I think you tend to use more specific forms of diagnostic criteria in low prevalence populations. We want to have more evidence that somebody has an unusual condition, whereas in high prevalence populations you want to be more sensitive. I think also in terms of the clinical application, one of the issues to think about is what are the costs of over-treating scabies versus under-treating. Scabies is a one-off or two-off treatment, has very few side effects generally, doesn't cost a lot, can make a big difference. So I think I would suggest to clinicians to use criteria that are more sensitive than specific, because I think the costs of missing scabies are greater than the costs of over-treating scabies. But it's just a couple of comments I'd have on that part.
During lunch we can continue this, but let's have this question as well.
Oh, it sort of relates to both of the previous discussions. So about half of the common samples had one cycle or more, right? Had one nanoplate, whatever. Sorry, my immunology is a bit basic. So that suggests that the suspect and clinical definition could be oversensitive, right? Because it looks like your test is really specific. So I guess the question for me is how do we haven't resolved the sensitivity question, have we? Because we're either saying they're over-diagnosing it with their suspect and clinical stuff, or that your test isn't sensitive enough, which seems bizarre because you're picking up one cycle in thousands. So, yeah, I think it's going to be really important for that further clinical validation. That's my, have I understood that right?
Yeah, it's correct.
Well, I think we've got some students doing a little bit of work on the agreement between the clinical findings and the PCR. And generally there's a reasonably high level of agreement, but it does suggest that given that information, that it's probably from a sort of population viewpoint, disease control viewpoint, better to overtreat rather than undertreat. We would suggest relaxing the IACS criteria to encompass some of the cases that Cielo's picked up. I think in terms of these PCR, we really need to have a good look at which ones are positive, do they correlate with the clinical symptoms, that sort of stuff. But I think it's a very exciting possibility because of the potential for being a very sensitive, being a more sensitive test than the qPCR is currently.
Yeah, but the 6 samples sent from Tonga were clinically suspect, we think. So it's not like they were...
the 6... the 6 samples were... they were... there were some cases that they were not part of the school's study. They were various clinical query cases. Some difficult cases that the hospital had asked me to collect some samples on. So they were tricky cases. They weren't barn door clinical scabies. The later ones were, we actually, in the last study that we did, we're thinking critically about the PCR test. So we're swabbing both normal skin and scabies lesions to try and see the correlation between the PCR test results and the scabies lesions. In the earlier study, we just swabbed positive lesions. But I think there's certainly more work that could be done on this.
But the case control, the second study, you can swab. You have controls, right?
Yeah, we have swabs from subjects with normal skin, what we thought were clinically normal skin.
So the second study will sort because they have controls.
That's the process.
I think we'd better move on.
Ladies and gentlemen.